Hello, BISC 210 students. Today I'm going to be covering Chapter 10, Toxins. So please make sure you read the PowerPoint as well as watch this video. It's going to be very important because there's quite a bit of important information in this chapter. I have significantly condensed it to the most important information, so that's what you need to focus on, whatever I cover. If I don't cover it, you don't. All right, so let's talk about toxins. So a couple definitions. Toxins, really, what are they? They're these molecules that are created by pathogens that can have an adverse effect to the host. Now, the effect to the host can be very, very different. It could be simply affecting tissue damage. In this case, also gives you an example of suppression of the immune response, or it could attack specific target cells. There are a lot of things that can happen within the broad scope of what toxins do. And we're going to talk a little bit about that scope in a few minutes. Something is toxigenic. That means it's a microbe that makes toxins. Toxemia means you have toxins in the bloodstream. And then there are two major classes of toxins, endo and exotoxins. Now, they're going to seem a little different when we know the term endo means in and exo means out. But when we look at them in terms of microbes, the way they look doesn't seem to line up with the actual terms. So what I mean by that, I'll show you in a few slides an image of a couple bacteria and toxins associated with them. So this is table 10.2 in your book. You absolutely 100% should go through this and go read it, practice it, because there's some really important information. Now, some of the big points here are, what are the toxins made of? If you're an endotoxin, it's lipid-based. If you're an exotoxin, it's protein-based. The made by, whether it's gram-positive or gram-negative, that can vary. Endotoxins are made by gram-negative bacteria because they have an LPS, which you guys should remember that the LPS is the lipopolysaccharide layer, right? Where exotoxins are produced by both gram-negative and gram-positive. So when it comes to exotoxins, there's a bigger range of organisms that can create them. We'll get to that later. Released from, that's something else that's really important. Gram-negative, as I just mentioned, the LPS contains the endotoxin. So when the bacteria goes through binary fission and it breaks the cell wall, the LPS layer, it will release some of those toxins. Or if the cell dies, it will release those toxins when the LPS layer breaks up. So the gram-negative cell wall, once it's destroyed in one way or another, whether that's through using antibiotics or even just natural binary fission, the toxins will get released. Exotoxins, on the other hand, are made inside the cell, and then they are released out into the environment. Now, they're part of the normal metabolism. So basically, an exotoxin is just something they make as part of their normal metabolic functions. However, they secrete them. Now, this is a good example, for instance, with Staph aureus. Staph aureus and causing food poisoning, food intoxication. What happens is it produces all these toxins and it secretes them. And it's not the fact that you eat, let's say it's on a piece of lettuce. It's not the fact that you eat the pathogen, the Staph aureus. It's the fact that you're consuming all of the toxins, the exotoxins that it's been secreting over its lifespan. So it's important to know that the actively growing bacteria, part of their metabolic function is to produce exotoxins. Vaccines, endotoxins, no. And exotoxins, some of them do have vaccines. We'll talk more about vaccines in another chapter. Can it be neutralized? No, for endotoxins, and then for exotoxins, some. Again, we will touch on that a little bit more later on. So if we look at endotoxins first, we mentioned that the LPS layer is its outermost membrane, right? The lipopolysaccharide layer. And the LPS is made up of quite a few things. We've kind of gone through its structure before in previous chapters, but the big components are a lipid portion and a bunch of sugars, right? A lipid, polysaccharide, many sugars. So the major components are lipids and sugars. Now, the lipid A region of the LPS is called the endotoxin, which is toxin to us and other animals. Now, when I show you how it functions, you'll see that there's a lipid A portion and then there's a B portion, and they have different functionalities. So here's a perfect example of an endotoxin. So here on the left you have a gram-negative bacteria. This is the LPS layer and its outermost layer. All of a sudden, the cell wall is breaking apart, and it is secreting all of those endotoxins away from the cell. So it's when the cell wall, the LPS layer, breaks apart that the endotoxins are actually released. Now, there's something to keep in mind with this. If you have an organism that's a gram-negative pathogen that produces an endotoxin, and you give a patient antibiotics that attacks the cell, the pathogen cell, could be the cell wall, could be any other function. If it kills the cell, all those endotoxins are going to be released. So in turn, what can actually happen is by giving a patient antibiotics and killing the pathogen, it can make that person even more sick than they already are, because all of a sudden their body is going to be flooded with endotoxins. Okay, so sometimes antibiotics are not the most ideal ideal response. Sometimes, depending upon the pathogen, it just may be waited out. Or others, we use specific antibiotics that don't actually kill the bacteria. What I'm referring to are antibiotics that are what we call bacteriostatic. Now, bacteriostatic simply means they inhibit the pathogen from continuing to grow. So that makes a lot of sense to give a patient with a gram-negative bacterial infection that produces an endotoxin. It makes a lot of sense to give them an antibiotic that is bacteriostatic, because that bacteriostatic antibiotic is going to prevent the pathogen from continuing to grow, continuing to really function, but it's not going to destroy the LPS layer and release the toxin, the endotoxin. So it kind of just inhibits its ability to function. Where if you give somebody a bactericidal antibiotic, that antibiotic kills the cell. It comes in and destroys it. And that's where you can have a problem with some of these gram-negative bacteria that produce endotoxins, because if you give somebody a bactericidal antibiotic, then it's going to kill the pathogen and it's going to release more endotoxins than it normally would if it were alive. So this is why we don't blindly just give one antibiotic for everything, because there are different situations that call for different ways to treat that person. The immune system is pretty good at getting rid of the toxins. It's also really good at getting rid of pathogens. But again, killing the gram-negative organism releases more pathogen, or more toxin, excuse me. Endotoxemia just means, remember we said toxemia is talking about toxin in the blood. This is more specific. This is endotoxins in the bloodstream. And then eventually a person could go into septic shock if it's a high enough concentration. Now, with septic shock, has some really unique symptoms to it. So it's a very serious condition that's typically what we refer to as a systemic infection, meaning the whole body, system-wide infection. And a lot of the symptoms associated with that could be organ failure, really low blood pressure, fatigue, low body temperature, tachycardia, and possibly even rapid breathing, what we call tachypnea. Okay, so septic shock is a really severe case. If you have too much of that endotoxin, a person can slip into septic shock and they can die. It's not an overly common problem. It's a little more on the rare side that it gets endotoxemia gets so bad that it ends up as septic shock, but it can absolutely happen. Treating them, you can't really neutralize them, and there's no vaccines to protect against them. Basically what they do is they treat the symptoms and they try to flush your body out of the toxins. Lots of fluids, electrolytes, and then medication to basically curb some of the symptoms that you're actually experiencing. So it's important to understand that endotoxins are released when the LPS layer is broken up, and that it's a very difficult mechanism to handle or treat. Okay, so exotoxins. Exotoxins, a little bit different. Remember, they are protein-based. They're obviously toxic. They're soluble. The effect of exotoxins is very wide, and again, both gram-positive and gram-negative bacteria can produce exotoxins. And there are three major classes that I'm going to expect you to know, and one example from each class. So we'll get to that in a minute. So how do we name exotoxins? We name them based on what it targets. So neurotoxin affects the nervous system. Enterotoxin, GI system. Hepato is the liver. Nephro is the kidneys. So we use these descriptive terms in order to pinpoint and say, okay, we have a hepatotoxin that's affecting the liver. And then you can look specifically for things that are happening to the liver and possible treatment. There are a lot of exotoxins out there. We've described a lot of them, and they're again classified into three major families based on their actions. And I do expect you to know all three types. However, I will only expect you to know one, maybe two examples from each family. So this is table 10.3 in your book. Everything highlighted in red is something you should know. So type 1, membrane-acting extracellular toxins. So basically that tells us a lot, that description. So the toxin acts on the membrane extracellularly, meaning on the outside of the membrane, and it can do quite a few things. With type 1, the one I really want you to know is superantigens, which are commonly caused or created, by strep and staph species. What's interesting about superantigens is that they attract the immune system, but they do so in a manner that's overwhelming to the body. So it's kind of like me standing up on a table in the middle of a dining hall and starting to yell gibberish. Okay, All of a sudden, everybody's staring at me. People are coming along to try and pull me off the table. I don't know why I'm on the table, but I'm on the table yelling. So they're trying to pull me down. That's essentially what the immune system sees. It says, "Oh wow, this thing is it's triggering an over response by the immune system." So not only does the toxin do damage, but it triggers the immune system to overreact and to do even more damage to our tissues. We'll talk about superantigens specifically in certain specific pathogens later in the semester. But for right now, just knowing what a superantigen is very important, and that it is a type 1 membrane-acting extracellular toxin. So triggers an overreaction by the immune system, which ends up harming you. Type 2, membrane damaging, so hemolysins, which you all should know very well what a hemolysin is. We talked about in lab hemolytic reactions. So a hemolytic toxin kills red blood cells or white blood cells and can interfere with our immune response. Specifically in lab, you talked about beta hemolytic colonies produced by Staphylococcus aureus. Okay, so we actually were looking for Staph aureus when we did our nasal swabs, right? And we plated it on blood agar. And plating it on blood agar, what we did is we said, okay, if this organism is present, then we would see a hemolytic reaction killing the red blood cells, and that indicates an organism that makes this type of toxin, a hemolysin. And we were looking at specifically, do you guys carry Staph aureus as part of your normal microbiota in your nose, which I am a healthy carrier. So even when when I'm healthy, I still have it in my nasal cavities, in my nasopharyngeal region, and when I plate it on blood agar, it grows. Remember back to that lab that we talked about 3 types of hemolytic reactions. There was alpha, beta, and gamma. Remember, alpha is partial breakdown of the red blood cells, and when you look at them on a blood agar plate, they look greenish in color. Beta hemolytic reactions, that's where the red blood cell is completely broken down. The red blood cell is destroyed. So what you can do is you can take your plate and hold it up against the light, and if you can see through the plate, through the media, then there's absolutely beta hemolysis occurring. And then gamma, there's no breakdown of red blood cells, but there is growth on the blood agar plate. So, and the colonies usually look like white to grayish in color. So those are type 2 membrane damaging toxins. Type 3, intracellular toxins. Remember, intra means inside. So these are ones that are inside the cell. The 2 I expect you to know are pertussis toxin, or what we call the PTX toxin. It's a cytotoxin made by Bordetella pertussis, which causes whooping cough. And it suppresses the host immune response. Now, I know right now a lot of this doesn't mean much, but when we get to diseases of the respiratory system, we're going to talk about pertussis at length and the PTX toxin. But for now, it's just important to know that it's cytotoxic, and basically it suppresses host immune response. Cholera toxin, you've probably heard of cholera before. The organism is Vibrio cholera, which causes diarrhea. It's an enterotoxin, which means it affects the GI tract. Okay, it affects the GI tract. These are all intracellular, meaning inside the cell toxins, so they're pretty damaging. We will talk more about the PTX toxin later in the semester. But those are the 2 examples I expect you to know for type 3. So I could say in a test question, superantigens are a type 1, 2, or 3 exotoxin. Or I could say Type 1 is considered what? Membrane-acting extracellular toxins. Or I could say, what is type 3? And you would have to say intracellular toxins. Or give me an example of a intracellular type 3 toxin. You would tell me the pertussis toxin or PTX toxin. So it's really important that you guys understand this table and go over it and over it and over it as many times as necessary to remember which ones go with which. So here's a little building on my superantigen example. Overstimulating the immune system, causing massive inflammation. The superantigens are going to bind to receptors on the surface of our cells. And really what it is it triggers a signaling cascade. So it triggers a bunch of chemicals to start to act once it binds to the receptor. And when these signals act, it actually changes gene expression. And the more of a change you get, the stronger the response usually is from the immune system, which can again damage your own body because they're overstimulating it, the immune system. Overstimulation can lead to damage. Now, we will talk about another organism later in the semester, again a respiratory organism, Bordetella pertussis, and then we'll talk about Mycobacterium tuberculosis, which both trigger the damage created by the immune system. So, again, superantigens, very important. Type 2, membrane damaging. Again, hemolysins. We talked about this, just talked about it a minute ago. Damages the membrane. The cell will rupture either partially or completely, depending upon whether you have alpha hemolytic or beta hemolytic hemolysins or hemolytic reactions. Cholera or pertussis, which pertussis we call the PTX toxin. Intracellular, this is what we call an AB toxin, an AB exotoxin specifically. The A portion is the active portion, which triggers an effect inside the cell. The B portion is the binding portion. So you have the A portion attached to the binding portion, the B portion. They attach to the cell, the target cell. And the binding portion will allow for the active portion to enter into the cell and cause whatever it affects within that cell, whatever the effects may be. And the effects that occur are pretty wide range depending upon what toxin you're looking at. But again, the active portion enters the cell and triggers some type of reaction. This, I don't have the image number on this. I believe it's somewhere later in the chapter. See if I can find it. It is figure 10.4. It just shows you what we've been talking about in an image. The first one is a superantigen, right? It binds to, it's a type 1, binds to the receptor, triggers an overreaction of the immune system. Type 2, that's the middle one here, that's causing hemolysis, right? Breaking the cell membrane down. Okay, the cell membrane starts to break down. And then the last one is the AB portion, or the AB exotoxin. You see here how the binding portion attaches to the membrane and it brings the active portion with it. And when it enters the cell, you have both portions in there, the A and the B. The A goes to work, and the B will kind of just hang out, or sometimes it'll be released. Depends upon the toxin you're looking at. So how do they hide from the immune system? I mean, right, we're talking about all these pathogens that can do these crazy things with toxins. So why aren't we sick all day, every day, right? How is it our body can manage to protect us? Um, well, our immune system is really strong for those people that are healthy individuals, um, but sometimes pathogens are really good at evading host immune defenses. And there are three examples I'm going to talk about in a second, but they're really good at uh, pr- uh, protecting themselves against the immune system and preventing the immune system from doing its job. So, antigenic masking. So, we talked about how an antigen, which we talked about in the previous chapter, is something that sticks off the pathogen off of its cell wall, or cell membrane if it doesn't have a cell wall, that's foreign to the host's body. This is what the immune system looks for. It looks for foreign antigens, things that stick off the cell wall. A good example, analogy would be scanning a code at Walmart, right? You probably have gone through the self-checkout before where you put something and you scan the barcode, and if it recognizes it, it lets you put it in the bag and pay for it and walk out. Well, you have to imagine if all of a sudden you had an item, you scan it, and it doesn't recognize the barcode, right? An error message will pop up. Well, in our bodies, instead of an error message, it sends a signal to our immune system and says, hey, this is not supposed to be here. It needs to go away. And the immune system will react and try to destroy it. But in antigenic masking, basically it hides its features, its antigenic features, by coating itself with host molecules. So now it looks similar to the original host molecule or the original barcode, where the barcode essentially is close enough to the original that's supposed to be there that our immune system will ignore it. So, original barcode, immune system goes off and says, "Whoa, hey, we need to get rid of this thing we don't recognize." But if it coats it in host molecules that the immune system does recognize, then it'll kind of just pass by it and it really won't have an issue. Antigenic mimicry. So it acts as if it's the host. It emulates the host molecules. Capsules, for instance, can resemble carbohydrates that stick off of our cells. So our cell membranes, every cell we have has things sticking off of it. Now, almost every single cell. Now, what they do with antigenic mimicry is essentially copy that. And by copying that, the immune system gets tricked, and it doesn't think that that pathogen is harmful, and it thinks it's part of the normal body system, and it just goes on looking for other non-host cells. Lastly is antigenic variation. This is a really interesting one. Because what happens is these surface molecules that are sticking off the cell walls or the cell membranes, in this case, can actually change, which is quite impressive. The flu virus actually has what we call antigenic variation. It has these spikes that stick off of it, and it can change from one version of the spike to a different version. Now, what the problem is with this is our immune system recognizes, let's say, it recognizes the original surface molecule, the original antigen from the disease-causing microbe, the pathogen. It recognizes that and it says, okay, I need to fight this. We need to mount an immune response against this pathogen with this given antigen. But if all of a sudden, which can happen, the pathogen changes its antigen that's sticking off the surface, you're going to have your immune system kind of stall. It's going to say, wait a second, I don't recognize this. There was something else here before that I do recognize that I'm ready to fight and get rid of, but now there's something different. And it's almost like the immune system has to start all over, because when it recognizes a foreign antigen, all right, it triggers memory B cells, memory T cells, cytotoxic T cells, antibodies, triggers production of all these cells against that known antigen. But then all of a sudden, if that antigen shifts from being seen by the immune system to something it's never seen before, well, then you have a huge problem, because you started building all that immune response to the first antigen. But now that this organism has altered it to a different variation of it, your immune system now has to go back to the drawing board and start building up B cells and T cells and antibodies again for the second antigen that was on the surface. So it kind of delays the immune response, because the immune system has to start all over against the unknown antigen. So this is a really, really useful tool for pathogens, and we will talk about antigenic variation when we talk about the flu, the influenza virus. So I like this image. Hiding from the host defenses. It's talking about masking in the top one, right? It covers itself in host factors, and the body thinks it's a normal cell. Mimicry, right? It copies the host molecules. You can see it's got a nice little top hat here. And then lastly, variation. This is what the immune system sees the first time, and it's getting ready to fight this specific antigen that looks like this, and then all of a sudden, it shifts, and now the antigen looks like this. And because of that shift, when the immune system comes along and says, okay, let's fight this first antigen, it's no longer there. The antigen looks completely different than it did when they first started developing T cells and B cells and antibodies. So now your immune system has to start all over to rebuild more B cells, T cells, antibodies to this version of the antigen right here. Okay, so very interesting. We do talk about pathogens that can do these things later in the semester when we go through the diseases of the organ system. We talk about individual pathogens. Other ways to undermine the host response. Our cells can be evaded by interfering with phagocytosis, or even suppressing the immune response in general, preventing certain chemicals from being made. Phagocytosis, if you recall, is when a cell engulfs a pathogen and it goes into the cell, into the white blood cell, and it destroys the pathogen using enzymes to neutralize it. So some examples of interference with phagocytosis. Phagocytes are going to engulf and destroy using enzymes, as I mentioned, but some pathogens can avoid phagocytosis by making a capsule, bursting free of the phagosome. The phagosome is kind of a bubble inside of the white blood cell that has specific enzymes in it. And typically the phagosome fuses with a lysosome. Okay, and when it fuses, it's able to effectively use a bunch of different enzymes to destroy that pathogen. But some pathogens can block that fusion, neutralizing the enzymes in the phagocytes. There are some pathogens that don't care if they get phagocytosed and they get put into either a phagosome or a phagolysosome, what they do is they just neutralize the enzymes and they continue to grow. We'll talk about pathogens in the GI system that can do this. For instance, Shigella and Salmonella can neutralize these enzymes. And then damaging phagocytic cells using toxins. Remember, toxins have specific targets in many cases, not all cases, but many, and they can attack specific cell types. So these toxins that are being produced by the pathogen can attack phagocytic cells, specific phagocytic cells. Here's a kind of what I mentioned a second ago, is that some pathogens can thrive inside the phagolysosome. They can just do perfectly fine in there because they can in most cases neutralize the enzymes that would normally destroy other pathogens. And we will talk about several that can do this. And this is something that's very serious, because once the organism is inside your cell, inside your white blood cell, and it's inside the phagolysosome, our body is counting on its ability to destroy that pathogen. And in some cases, we can't do it. Our immune system is kind of blocked in terms of the phagolysosome being effective in destroying a pathogen. Here are some images that I wanted to show you associated with phagocytosis. Here on the left, this is what happens in phagocytosis. The white blood cell, the phagocyte, engulfs the pathogen. This is going to be the phagosome. Here's the lysosome that has the enzymes in it that destroy the pathogen. They combine to create the phagolysosome, and the enzymes chop up the pathogen and just destroy it. But on the right here shows how pathogens have learned to evade phagocytosis, releasing toxins that kill the phagocytes. The capsule prevents what we call opsonization, which is when the phagocyte engulfs the pathogen. It can actually prevent that. Here we have the blocking of the fusion of the phagolysosome. So remember, the lysosome has the enzymes, the phagosome has the pathogen. So if you, in a healthy phagocyte, they combine and the enzymes are released into the phagosome, whereas in this case, right here, they block that fusion so the enzymes don't get into the phagosome. Here we have cells escaping the phagosome into the host cell, the host white blood cell, and it's perfectly fine in there. And then lastly down here, adapting or neutralizing the enzymes that would typically destroy a pathogen. Here, even though the phagolysosome is created, those enzymes that are secreted in there in the phagolysosome are not effective in destroying the pathogen. Okay, all of these really good examples. I like this image. I think it's very useful. I would recommend looking over this and making sure you understand each example. Immune suppression. So suppressing the immune system is another way for pathogens to just run rampant and not be controlled. Basically, they can target our own immune cells, so it's kind of like a preemptive strike, so to speak. Before the immune system can make all the cells it needs to fight the pathogen goes and targets the immune system cells on purpose, and it starts killing them before they can actually attack the pathogen. Antibodies, which are these proteins that we make to attach to antigens, and antibodies have several functions. One of the functions is when they attach to antigens, they make the pathogen look bigger, because remember, bacterial cells are much, much, much smaller than our cells. So they can kind of go unnoticed sometimes by our immune system. So what our antibodies do is they attach to a specific antigen, and when they attach, it makes the cell look bigger, so the immune cells can recognize it and say, oh wait, okay, that's not supposed to be there. Antibodies can also neutralize toxins in some cases, not all, but in some. Antibodies can neutralize toxins. But bacteria can make proteases. Remember, if it ends in ase, it's usually an enzyme. So a protease is an enzyme that specifically breaks down antibodies. So there's one way. Interleukins are chemical signals associated with the immune system. If the bacteria can interfere with the production of interleukins, a good example would be cytokines, those communication molecules don't get sent, or if they get sent to the wrong place, or they're non-functioning, they're not correct. So interfering with chemical signals associated with the immune response is another way to suppress the immune system. And then interfere with signaling. That is absolutely one of the major ones. Is our cells send chemical signals all over the place. They talk to each other with chemical signals. They activate the immune system. They activate defenses by using chemical and molecular signaling. But if the pathogen can interfere with that signaling, then that part of the immune system is just not going to function. And by not functioning, that pathogen will have the ability to continue to grow and increase in numbers. And the infection becomes much worse after that. Now, there's one term that is not in this PowerPoint that's posted on Blackboard, but I'm going to post this one slide. It's called the term is called tropism. And tropism is an example that, or is a term that describes that a specific pathogen prefers a specific host, and in some cases, a specific tissue within that specific host. So tropism, just talking about a pathogen looking for a specific host. Good example is Shigella flexneri. So this organism likes to attack our GI system. It causes dysentery, and the pathogen attacks the gastrointestinal tract, specifically in the intestines of humans and primates. So this organism is a really good example of tropism, where that pathogen is unique to humans and primates, right? And then specifically it looks for places to colonize and cause infection within GI tissue, gastrointestinal tissues. So again, tropism is a really important term, talking about how specific a pathogen can be, not just specific to a particular host, but also to potentially even more specific, a specific tissue within a specific host. So I'm going to add this slide to Blackboard as well after I post this video. So I think this is the last slide. I know I condensed Chapter 10 kind of significantly, but I expect you to know what we talked about. Anything I didn't cover, you are not responsible. So if you have questions, please email me, let me know, and I will try to help you as best as I can. Everyone stay safe, and I will talk to you all soon. Have a nice night.